part. So this afternoon we'll talk about phytoplankton. So we will see, we will have more or less the same schedule than items. So there is some basic things. So I will go quite fast for this. And then I will leave the floor to Clarisse and to Benjamin. So for this first part, it's still some reminders. Oops, sorry, free tech. Some reminders about the biology and the diversity of phytoplankton, and then second point about classical methodology for phytoplankton biomonitoring. So very briefly, some definitions, a few things about their diversity, endosymbiosis, some reminders still, and a few things about phylogeny and taxonomy. So these are very basic definition, but I think I can skip. Just to say these are very simple organism and even more simple when you consider cyanobacteria and when you compare to higher plants. So there is a huge diversity of shapes, as you can see here. And just to have a brief look in the tree of life, where are they? So if you consider here prokaryotes, so in prokaryotes you have already here the cyanobacteria, which are the first chlorophycean organism which appear on the earth. And then here, this big group is the Chlorobionta, in which you have Chlorophyta, so an important group of algae, Streptophyta, in which you also have some algae, and Embryophyta, which are higher plants. Yes, this is for you, Alexandra. And more basal, you have Rhodophyta and Glaucophyta, quite near this Chlorobionta. Here you have animals, two fungi. So what else? Cryptophyta. Here we have euglenids, Euglenophyta and Euglenozoa. Here Aptophyta, an important group also in freshwaters, not only in marine. And in Heteroconta, you have this group, which is the Heterocontophyta. And you can find again our diatoms besides Zontophyceae, Stigmatophyceae, and Chrysophyceae. And the last group are Alveolata with the Dinophyta. So yes, this is a completely, when we talk about algae, it's completely polyphyletic. And this is explained by endosymbiosis. Just a reminder of endosymbiosis and the different endosymbiosis we have and the groups it gave. We have primary endosymbiosis with, let's think about, this is a cyanobacteria with this circular genomes. It has been endosymbioted by a protist. Here, this is its nucleus. And let's say that the X are the chromosome. This gave mostly, so we had gene transfer, and this gave mostly the Glaucophyta, Chlorophyta, and Rhodophyta. And when you look, of course, when you look at the chloroplast of these groups, you have membranes coming from this membrane of the cyanobacteria and the membrane of the protist. So this is the primary endosymbiosis. So now let's, and again, you have you had a secondary endosymbiosis with a protist. So you take a eukaryote algae. Again, you had gene transfer between both organisms, and this became the chloroplast of, for instance, diatoms, so Heterocontophyta, Euglenophyta. So here you can have 3 or 4 membranes because one membrane disappeared with the evolution for some of the groups. And you can have in some chloroplasts of some groups what we call a nucleomorph, which is the remainings of the nucleus of for instance, the Chlorophyta. And we had, in some cases, a tertiary endosymbiosis. So again, same process, and this gave mostly the Dinophyta. So I already talked about this paper of Adel with this tree, and just to give a picture of different endosymbiosis, we started with a primary endosymbiosis from a cyanobacteria. It gave the Glaucophyta, for instance, here, the Rhodophyta, and the Chlorophyceae. secondary endosymbiosis. So you start with Chlorophyta, and it gave the Euglenozoa, Euglenophyta here, or from the Rhodophyta, it gave the Aptophysi, the Cryptophysi, and here all this big group with the diatoms, the Chrysophysi, Bolidophysi, and Xotophysi. And the tertiary endosymbiosis, mostly for dinoflagellate, their chloroplasts are coming from Rhodophysi, but also diatoms and Aptophysi. Okay. So it's reminders, I think. An overview of the main algal groups. So here I will go a little bit fast, but just to give you a few pictures of what are cyanobacteria, so you can see that they can be filamentous, some are unicells, what else, some are making some strange colonies like this Merismopedia and on. Some are producing some akinets. So akinets are in fact resting spores. Others are making these heterocysts, so to have a nitrate fixation. And some have aerotops, like these small gas vesicles here. Okay, these are the chlorobionta. So this is a short phylogeny of this group, where you can find here higher plants. This is for you, Alexandra, where you have, but no, the Charophysi are here, sorry. So you have the Chlorobionta, two groups, the Chlorophyta. I put for you a nice video. Are you happy? You can see the diversity that we can have. So I think these are, sorry, help me please, Chlamydomonas. We can jump to the other group of Streptophyta. I gave here a few pictures of this group, Zygnematophysi. Also, very strange shapes. Some are being some being filamentous over unicells. A very big number of species. Again, a nice video of Euglenophyta. You can see the number of chloroplasts it has here. Euglenophyta are usually living in very high organic matter concentrations. What else? Okay, yeah, maybe I can skip. Chrysophyta, on the contrary, so you can see the number of species, very different shapes. Most of them are living in low nutrients level, like these dinobrions. There are several species of it. And diatoms, 3,000 species. Its characteristic is to have these plates, so an extracellular skeleton in cellulose, and these strange plates. You have several toxic algae in marine environment, but not in fresh waters. Okay, some brief things about phytoplankton biomonitoring in lakes. Into what we are doing in France, we use this index, which is called the IPLAC, and its aim is really to assess the trophic level, so the nutrient concentration. And so this is the first aim, and the second aim is really to be representative of the overall lake water quality. So to be representative of what can happen in all the lakes. So there is a paper of a very good colleague, Christophe Laplace, who is in our project with Benjamin, Agnès, and myself, where we are developing this metabarcoding for phytoplankton biomonitoring. So he published, it was, yes, in 2016, this IPLAC. And the lakes which were used to construct this index. I will talk about these 4 points in the next slides. First, how we make field sampling. the lab procedure for microscopy, and then you will be able to compare to what Clarisse will present you for DNA metabarcoding. The estimations of the biovolume, which are important points for phytoplankton, and the metric calculations of the IPLAC. So field sampling. Usually, not usually, that's the rule. The position of the sampling site must be, you must avoid these areas. And the springs of the deepest points in dams is here near the dam, in fact, near the wall. You have to avoid this area because there are some algae which are developing on this wall. And to avoid the contamination, you have to be from a certain distance, which is enough to avoid this contamination. So you have to avoid these littoral and benthic species, which are not part of the phytoplankton. These are the devices that you may use for phytoplankton sampling. About the sampling frequency. For the WFD, for the Water Framework Directive in France, there are 4 samplings per year, and this is carried out every 6 years in all the lakes in France. So we don't sample every 6 years all the lakes in France, but there is, how do you say, a roulement, a turning. So we start with one region, then with a second, and every 6 years, all the lakes have been sampled. And basically, we sample one sample every season, one during winter, one during spring, summer, and at the end of the summer stratification, so just before the autumn. The sampling layer, which is sampled, is the euphotic zone. So just to remind you what is the euphotic zone, it's the layer that extends to a depth residual light of about 1% of surface light. So it's between the surface and the depth where you have something like 1% of the light. And to measure the euphotic zone, what we use is the Secchi depth. Do you know what is the Secchi depth? This disk, which is white and black. Okay, I see everybody say yes. And we use an integrated water sampler. It's this kind of bell, what we call a bell, because it has this shape of bell, which collect regularly the water with the depth. So the water enters into this device proportionally with the depth. So we can use some mechanical samplers like this, which is based really on the shape of the bell and the pressure, or something which is programmable with there is a small microprocessor, an electric engine, and it's like a syringe which lets the water enter this tube progressively when the pressure, the depth is increasing. Okay. So this is in the field. So you come back to the lab and you're going to look at it on the microscope. We use this kind of sedimentation column. So we take this is the sample. It's yellow because of the lugol. So it's a way to preserve the sample. And you pour this sample in the sedimentation column during a couple of hours. So you do that, you put all your sample here, for instance, 25 milliliters. The algae sediment settle down on this small slide, and then so you remove the column and you look at it on the microscope. So under the microscope, you have not at 100, but at times 40 usually, and you count 400 cells or algal objects for a known volume of sample. And for each cell, the specific biovolume has to be known. So for instance, for this chlorella, you must know the biovolume of one cell. and then you have to multiply the number of cells per milliliter by this volume to have for the community the full biovolume of your community, and you even know for each species the biovolume of each species per milliliter. Okay, estimation of biovolumes, that's just what I said. So each cell you've seen in the photos have a different shape. So you have to assign to each cell a geometric shape. For instance, this Mujosia gracillima cell, the nearest geometric shape is a tube. So there is a formula of a tube that you can find, for instance, on Google. You ask Google, find me the calculation of a tube. And so, for instance, this one is a little bit more complex. You put several cones for each arm, and so on and so on. So really, the final result of a floristic list is this. You have some species names, and for each species name, for instance, this Diatoma elongatum, this first result, it's a number of cells per milliliter that is transformed into a biovolume of algae per milliliter of sample. So I relate it in in cubic micrometers of algae per milliliter, but you can transform it in something different. But basically, that's it. Metric calculation. How do we calculate our IP lag? But just before, but I don't know if it's necessary to enter into such detail. But just to be aware for you that in a lake, the communities of algae are changing from a month to another. This is, for instance, in Lake Geneva in 2010, you have the different months of a year from January to December. And so every month we have a sample which has been taken. So we make the countings and the determination. And here you have a biomass. So you can see that the biomass of algae are changing, and each color is corresponding to a different class. So you can see that in winter, in spring, summer, and autumn, we don't have the same communities. And this is why for the IP lag, we need these 4 different samples per year to have a correct evaluation of the community during in the lake. So for instance, this was in 2010, but you can see that in 2009, it was a different seasonal dynamic. And so these dynamics are changing from a year to another. And here it was last year. So you see again a different dynamic, a seasonal dynamic. So just to be sure that we are on the same line, it's important to capture this seasonal variability, but also this interannual variability. You saw that in 2010, it was not the same community that in 2012. So we have to collect all this variability. And this is why we have these samplings for the IP lag, which are carried out every 6 years. So the IP lag is based on the phytoplankton communities. So spring, summer, autumn. Even if we have 4 samplings, we only use 3 seasons. And yes, the IP lag is measured every 6 years. We could make measurement every year, but it costs some money also. So it's a threshold between what we can do and the money available. There are 2 metrics which were calculated in the IP lag: the specific composition and the algal biomass. So the the metric of specific composition, you know it. It's the Zelinka and Marvan formula. So still the same story. You already saw that. Do you want me to repeat? No. Thank you. So you know it. This is a classical biotic index. The IP lag gathers the ecological profile of 165 taxa. And this metric for a year is in fact the average of the 3 metrics of spring, summer, and autumn. The second metric is the algal biomass, and it's based on the chlorophyll A concentration in the sample. And so this metric is the year metric is an average of the 3 metrics of spring, summer, and autumn. And the final calculation of the IPLAC, it's a weighted average between the specific composition, the metric of specific composition, and this algal biomass metric. And it gives more weight to the metric of specific composition than the chlorophyll A concentration. Okay, I skip this. So we have this IPLAC calculation. So the objective of using DNA metabarcoding, what is the objective of using DNA metabarcoding for phytoplankton biomonitoring? It's to try to calculate this IPLAC like we did for diatoms, but with DNA metabarcoding. So this first metric, the metric of algal biomass, I hope you understand that it's not possible to assess it with DNA metabarcoding. The chlorophyll A is analysis which is done in either with a probe or in chemistry. So we don't need to develop a methodology for this. We keep the classical way. But yes, really the objective is for this specific composition, because in fact it's quite challenging to get it with microscopy. You know, Milos, it's quite time consuming to make all these microscopic countings. Taxonomists are quite rare, at least in France, they are becoming really rare. And the objective is really we made the assumption that DNA metabarcoding can ease this analysis based on the experience we had with diatoms. And yeah, and that's all. So this was just really to set the objective of the next presentation. Do you understand the objective of what we are now working on with phytoplankton? And in fact, we started a project almost 3 years now, 3 years ago, to develop phytoplankton DNA metabarcoding for biomonitoring in lakes. Do you have questions? No? So maybe, sorry.
I have a question. When you said that you can use DNA metabarcoding for this MCS, can you explain that a bit more? So this is a species composition.
Yeah. Yeah.
But in a sense of the number, what would be the number? Okay, you have a different percentage of different taxa.
Yeah.
But can you explain a bit how it looks like, that number? I'm really not sure.
This number, I mean, you mean the
calculation, yes.
So the calculation of the MCS, it's this weighted average. So here is, sorry, I should change. It's not the abundance, it's the biomass. And so the biomass of a species that is multiplied by the indicator value that you find in the methodology of IPLAC. And again, this stenosis degree that you find in the IPLAC methodology. And in fact, using the biomass or the percentage in the Zelinka and Marvan formula, it's the same. I mean,
okay, so we have to replace the number that we would otherwise get with the counting,
yeah,
with the numbers that we will get with metabarcoding,
yeah,
and to multiply it with this indicator value and the
exactly.
weight, and that's
But
it.
the objective, you will see the result in a few moments.
Thank you.